The single cell is a biological machine powering essential functions from respiration to replication. However, the true marvel is the properties that emerge when cells communicate with each other. Cell-to-cell communication is a fundamental process that allows organisms to respond to a dynamic microenvironment. Disruptions to these complex signaling networks interfere with cells' abilities to function properly. Researchers study these processes because it reveals valuable information about cell behavior and disease. Cell communication is often achieved through protein secretion and membrane display. Currently, this communication is studied using genetic or protein circuits, but these tools lack the high spatial and temporal precision needed for targeted applications that require fast response times. Thus, there is a need for more sophisticated research tools to study intercellular communication. Here, we have developed Photocrete, a toolbox for light-inducible protein secretion that will help researchers overcome spatiotemporal limitations by integrating optogenetics into the protein secretion pathway. Photocrete expands on a protein circuit called release, which controls protein secretion by using an ER retention motif and an upstream cut site that is only cleaved upon introduction of an exogenous protease. Cleavage allows the protein of interest to continue down the secretory pathway from the ER to the Golgi body and finally to the plasma membrane, where it is either secreted or displayed. Photocrete makes this system optogenetic by replacing different components with photoactivatable proteins such as focal, which breaks when light is shown on it. These modifications allow us to control 3 different forms of cell-to-cell communication: secretion, shedding, and surface display. Our toolbox will offer researchers an unprecedented level of control to probe into intercellular communication. This will be extremely valuable for fields such as organismal development, neurobiology, immunology, and more.